Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor and your host. As we approach World Cancer Day on February 4th, we're taking a closer look at how cancer diagnostics are evolving, not just in theory, but in real-world clinical laboratories that are shaping the future of oncology care. In this episode's conversation, we bring together three experts from around the world who work at the intersection of technology, research, and routine diagnostics. Joining from Portugal, we have Jorge Lima from IPATIMUP, one of Europe's leading oncology centers with a strong research-driven diagnostic mission. And based in Italy, we have Bernard Okere, a field application scientist at MGI, which provides a full spectrum of digital equipment and systems for life science companies. And joining us from China is Nancy Yang, the International Senior Business Development Manager at GenePlus, which is a genetic testing and precision oncology company. We're going to explore how IPATIMUP is using MGI sequencing platforms for GenePlus's capture-based RNA and DNA panels to extend what cancer diagnostics can deliver today and prepared for what's coming up next. We'll talk about validation, automation, partnerships, and how labs can grow without compromising quality, all with an eye towards the future of cancer care. Jorge, Bernard, Nancy, thank you so much for coming on the show from all of these different places in the world we're coming from. It is quite an international episode. So I'm really looking forward to jumping into all this. Thanks for coming on, everybody. Jorge, it would be helpful, I think, for listeners that may not be familiar. How would you describe IPATIMUP's role within the European oncology landscape?
So hello, Kira, hello everyone who's listening to us. So we at IPATIMUP here in Portugal and also in Europe, we are a leading molecular diagnostics laboratory. So our main goal is to provide the best possible molecular diagnostics, of course based on DNA, based on RNA and other types of analysis. And we want to be like that here in Portugal, also moving forward to Europe and perhaps other parts of the world. But I think we have established ourselves in Portugal as the leading molecular diagnostics laboratory. So our major goal is that everyone, and we are working mostly in cancer, but also other diseases, but specifically in cancer, we want to bring the best possible molecular diagnostics so that every clinical decision in Portugal is made with the most information that it's out there available.
Yeah, it's so important. And how do research findings at IPATIMUP translate into changes into routine diagnostic workflows? How are you balancing innovation with that clinical responsibility when introducing new technologies? Because science and particularly genetics moves very fast with this tech, but we don't want to compromise that quality and everything that you're talking about.
Absolutely. It's very interesting that you make that question because actually, IPATIMUP started out as a research laboratory. So we made research in cancer, and the findings that we were having, we thought, okay, so we are making the findings, we are producing scientific papers, we're out there in the scientific community, but we thought, okay, we can do more with this. And then we started out making and creating some genetic testing. At that point, we made the Sanger sequencing and everything else. Mainly for germline molecular testing. But as we move forward, as we started studying other types of tumors, we started to define specific genes that were important to some diseases. And so we started out with our own custom-made, homemade molecular tests. And that was the basis of the molecular diagnostics laboratory with which we provided external services. So I guess that we can say that research is at our core, is at our basis for establishing ourselves. Now, we still have homemade, custom-made tests. We now have more and more testing that are provided by external providers. And we have the so-called NGS testing, the exome testing and everything. Of course, we have to have them delivered from companies with whom we work with. And so the interaction between ourselves and our providers is essential so that together we can work and create, well, what is there already, but also what we can build upon what is already known.
Yeah, it's so helpful, I think, as you said, that you're coming from that research background and building upon that, because I think that is a really strong point of the lab. What has led to the factors of choosing MGI sequencing platforms? Because there's various ones out there. How
Mhm.
did you land on MGI?
Well, we attend different meetings, different congresses. So we actually, in 2024, we were in AGBT meeting in Orlando, Florida, and so we got together with different people from different companies, and we found that MGI was making a terrific job. We also have a colleague of ours that was working that had worked with us at IPATIMUP and was working abroad in other laboratories, working with MGI. So we got to know MGI for their capacity for sequencing with great quality and with reasonable costs. So at that point, at the beginning, was that two issues. One is quality, and the other one is quality with reasonable costs. And so because we wanted to enlarge our scope of techniques and ways of sequencing either in the germline or in the somatic setting, we thought, okay, so let's start working with MGI to see how the equipment and how our validation and testing behave themselves according to what we have working with so far. And so when we started sequencing with the MGI machines, we found that so what was praised by MGI was actually true because everything worked really smoothly and the quality is there. And everything that we do either in the NGS panels and also exomes that we are also testing is working really, really well.
Bernard, coming from MGI, what do you think differentiates MGI platforms from other oncology diagnostics, and anything else to add on to what George was sharing, coming more from the client perspective?
Yeah, sure. I think that George said it all because quality for sure is the first thing we try to accomplish, and it's always good to see that customers are the first ones to realize that, analyzing their data and see that the quality is there. Cost efficiency, of course, is another thing that we strive for, and I would add the third key word is uh, be cost effective, of course, but also be flexible. Okay. So customers nowadays need to scale up, scale down, they change applications and they need to be flexible. And from MGI perspective, working with customers that eventually want to grow and change, it's also a challenge for us, but it's also rewarding seeing that the results come to an end. So Ipatim was a great example of this.
Yeah, I can add upon what Bernard was saying because we learned with the process that the working with MGI is very rewarding because we work really well together and our questions and our doubts are readily answered in a very clear way. And what we need, well, of course, it's not always possible. That's what it is. But at least, and every time, there is really interest in trying to solve all the questions that we have. So, and most of them are solved and are improved. And so, what we have as doubts and some things that we are not understanding very, very well during the process of validation or implementation, all those questions are answered very quickly. Practically everything is solved in a timely fashion. So it's it's a process, and it's been really rewarding and very 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 good to work with MGI in that sense.
And it sounds like it's not just setting up a lab with the sequencers and everything, but keeping that relationship going to have that support and the collaboration aspect. Bernard, could you speak to that part?
Yeah, sure. Yeah, it really starts way before the installation and way before the training. I'm part of the FAS team, and here in Europe, we support the customer from pre-sales. So it's very important to address the concerns and also the future perspective, because you have to be also long-sighted. Okay, just not see just what's happening over the next few months, but also having into perspective how the customer could develop their applications in the future. So this is very important to address this at early stage. And then, of course, the installation, the first training, the first run. So everyone is on top hype and happy for that. But what we try to really do in our support team is to make sure that the follow-up is there, because the next run is the most important run. So be with the customer, try new applications, scale up. This is all part of the support team. And this is also for us rewarding because we keep ourselves motivated. We have to face different applications, different solutions, and the customer is happy knowing that MGI is always there, not only for installation, but throughout all the workflow and the routine operations.
And part of that workflow is actually having gene panels to choose from and use. So Nancy, we got to bring you into the conversation. Can you explain the solution you're presenting and how this is helpful in the clinical market when it comes to genetic testing?
Yeah, so actually we work with MGI, our long-term partner. So we together provide the all-in-one automated and cost-effective NGS solutions for oncology molecular diagnostics. So which means more patients could benefit from our solutions. So the turnaround time could be achieved within 24 hours from sample to report. So which means a lot to the patients because the physicians could make a quicker treatment decisions and the patient could receive quicker treatment. So in terms of the sample type, we have our assays are for both tissue and the liquid biopsy samples. As you may know, for some patients, it's hard to get the tumor tissue samples. So the liquid biopsy samples is a good option for them. So in the end, no patients will be missed. So, and our ICs cover the whole journey of the patients. As for the biomarker testing, for the precision medicine, we have the medium panel called 188 panel. So this panel is not too big or not too small, which can meet the clinical needs very well. And we also have a big NGS panel for more than 1,000 genes, which is called the 1021 gene panel. So this panel is ideal for the patient, for the users who would like to have more comprehensive information. So for the fusion detection, we have both DNA and RNA-based panels. So as George mentioned just now, they also provide the DNA and RNA-based detection. So in a summary, so we provide the total NGS solutions to minimize the lab workflow and have more patients benefit from the precision medicine at affordable prices.
And I'm so curious, you mentioned the two panels, the 188 and then the 1021, which is over 1,000 for people just to take that number in. How did you and your team end up selecting and designing these two main panels, particularly for the European market? And how do you recommend one over another? Because 1,000 genes, I'm so curious, but also I would be a bit overwhelmed of looking at all of that information if I was in the lab.
Very good question. Thank you. So actually our panels are designed according to the clinical needs. So firstly, we use the capture-based technology, which means good sensitivity and specificity. The 188 gene panel includes the essential biomarkers recommended by the ESMO guidelines and also novel biomarkers. The same panel can be used for both tissue and liquid biopsy samples. So for the clinical lab, we strongly recommend this panel. But if the lab would like to have more comprehensive information, such as the TMB, the tumor mutational burden, so we recommend them, they use this big NGS panel, called the 1021 gene panel, which is the CGP, Comprehensive Genomic Profiling Panel, which is for pan-cancer and solid tumor.
And George, what has been your experience in using these panels Nancy's talking about from Gene Plus? Because there's the RNA, the DNA, and we're combining this. So you're using these panels from Gene Plus on MGI's sequencing platform. So how does this all come together from your experience actually in the lab?
So I think we started our collaboration with a year ago, more or less. So we designed our plan together with MGI and Gene Plus. We designed our plan that we wanted to move our routine in the different settings that we have. So meaning We have lots of samples from lung cancer. That's the most samples that we have. And then from other types of cancer. Have different settings where we want to apply different panels. So we wanted to move. So what we were doing up to a couple of months ago in the lung cancer cases was applying a 52 gene panel. And as I said previously, we want to provide the best possible. So that will be to have more genes so that the analysis can be more comprehensive. So our plan and what we already did was to move our routine of lung cancer testing from the gene panel that we were using to the gene panel from Gene Plus. So, and this was accomplished in November last year when we moved everything. So we are now running all our weekly lung cancer samples in one run of the 188 panel in the MGI machine. So, and it's working really smoothly with a couple of things that we still have to improve, but those are details and we will for sure improve. But as a whole, it turned out really well. And that's the setting where we already moved. Now, as Nancy was saying, the 1021 gene panel is very interesting for advanced cancer cases, whichever the tissue is. But those cases where the patient is still fit, but the treatment alternatives are not clear or they have already run out of alternatives. And so maybe with a comprehensive genomic profiling, we can find actionable genes, maybe off-label drugs, I think those will be the cases. But still, in those cases where it is still worthwhile pursuing an analysis for the patient, that's the situation where it makes sense to have a comprehensive genomic profiling. The other one is rare cancer cases or strange cancer cases where there is much to be known still. So a comprehensive approach also makes sense. And so that's where we are working already now. And that's the setting we are thinking of moving to the 1021, like Nancy was saying. And then the other setting is the testing and implementation of the RNA fusion panel, because fusions are getting more and more important in therapeutic terms. So the broader we have the approach to the fusion detection, the best. Now we have tested and validated the 1021 gene panel, and we are in the final steps of the testing and validation of the RNA fusion panel.
That's so exciting. RNA is so helpful, I think, in understanding and defining more of gene variants that we see and saying, okay, well, is this actually affecting the function of that and providing another data point when it comes to that? We've talked about Gene Plus, these panels being used on MGI's tech. Nancy, anything else to add to just how MGI sequencers work well with these panels, coming from your perspective of being from the company that's making the panels?
Yeah, very good question. Yeah, thank you. So our panels, including the panels we mentioned just now, 1021, 188, and a fusion, etc. Yeah, all our panels are optimized and dedicated to the MGI sequencing platform from the first beginning. Yeah, and to use our library on the MGI sequencers, no need a library adapter convention, which means save some steps, yeah, for the customers. I may say, yeah, so our solutions is one of the best oncology applications on the MGI platform. Yeah, talking about MGI platform, the DNB technology uses the DNA nanobots. It's called DNBS. Yeah, to effectively reduces the amplification and the sequencing errors and ensuring high data accuracy and lowering background noise in the downstream analysis. This is critically very important for the clinical testing, especially in the blood-based assays or the MRD testing. So the technology is very good technology.
Yeah, certainly. And George, what gave you the confidence to move these panels from validation into routine clinical use, and as you're starting to with RNA as well?
So we have many samples from the routine. So we picked a number of samples where we knew already the alterations that were present. And we run those same samples with the Gene Plus panels. And so we tested the specificity and sensitivity of the panels. and the results were excellent. So we already knew from Gene Plus and from other editions of the panels that the performance was really good. But we always need to make it in our own hands, the performance. And so it turned really well. And so the fact that there was already literature about it, and the fact that in our own hands the performance was great, that gave us the confidence to move and to change everything from the previous panels to the Gene Plus panels right now. I also think that the fact that the 188 panels is or will be shortly IVD branded, it's very important in the years to come, because this has been delayed for a couple of years now, but eventually it will come to being mandatory to have IVD tests. And so they already are, and then we can be in an excellent position in Portugal to be the first ones to provide an IVD NGS test.
It's so exciting. I mean, I think RNA is such a wonderful tool that I'm so thrilled so many companies around the world are really starting to take advantage of. Nancy, from your perspective, like, how does RNA sequencing complement this DNA-based testing, especially when thinking about the complex oncology cases that we've talked about a little bit?
For the fusion detection, so the ESMO guidelines recommend both DNA and RNA-based fusion detection, which are complementary to each other. So for the DNA testing itself, actually it forms the cornerstone of molecular diagnostics. But in complex cases, RNA testing serves as a critical complementary tool. It can visualize the active fusion genes, overexpressed oncogenes, and aberrant splicing events that may be missed by the DNA-based assays alone. For instance, in common cancer types, such as lung cancer or colorectal cancer, if DNA testing result is negative, the complementary RNA testing is recommended. So the studies have shown that adding RNA sequencing in such scenarios can increase the detection rate of the clinically actionable findings. For sarcomas or rare tumors, upfront combined DNA and RNA testing is generally suggested. The integrated DNA and RNA analysis represents the current gold standard strategy for achieving the most precise and comprehensive molecular profiling of tumors.
Yeah. And George, how has utilizing RNA sequencing so far within genetic testing changed your diagnostic confidence with those results? Are there certain samples that you found? I know Nancy just mentioned some, but are there certain ones that RNA sequencing tends to be more helpful than others?
Yeah, so we are using right now the DNA-based 188 panel. And so our strategy is that that's the starting point. And whenever the testing is positive for EGFR mutation, for KRAS mutation, whatever, then it ends the cases, because we do not expect to have a fusion driver plus an EGFR or KRAS driver. So if we have those mutations, then we consider the test closed, and we found the driver mutation, so that's it. Whenever the case is negative, like Nancy was saying, then it's highly recommended to perform the RNA, and the RNA yields an extra 10-20%, depends on the situations, of actionable alterations. And that's why it's very important, because it's broader in the sense that it will catch more types of fusions than the ones that are caught with a DNA only detection basis. So the RNA is very important, and the RNA panel, also the RNA sequencing that we are also making, because that's the only way you can find new fusions, because it's what it is, right? I mean, fusions, because of their nature, can be already known fusions, but there can be new fusions. If a constitutionally expressed gene is fused to the kinase domain of an oncogene, that we should expect that fusion is the driver, and it might be or not target. So as a whole, RNA-based detection is very important, either as a fusion panel, at least from the starting point, it's very important, or in the further steps, broad RNA sequencing approach can be also an important method.
And a big part of getting all of this done in a timely fashion
Mm-hmm.
is an automation aspect to this. So we can't be manually doing all of this because then we wouldn't have those tight turnaround times that we've mentioned. Bernard, from your perspective, why is automation so essential, especially for this high throughput oncology diagnostics in 2026?
Yeah, so as we said, we need to maintain consistency. We have turnaround time that become more and more important, and we are in a clinical setting. Robots are becoming mandatory, and I'm happy to see that also automation has been implemented in IPATIMUP, and also GenPlus is part of that. Of course, also MGI provides automation. And yes, it's important, but more than this, I think it's also good to have some flexibility. Okay, flexibility is what I'm mentioning the most during this podcast. But also, automation must be flexible. Okay, we have different type of samples, we have different numbers of samples, we have different requirements. We want to be able to change the protocol a little bit. So having automation, open automation that could be editable from customers and also from MGI ourselves, it's mandatory. This will allow customers to easily implement new applications, and this will benefit the customers and the end user and the patient, which is the most important subject in this case.
Every lab is different. So it's a lot of figuring out and customizing. It's not really like a plug and play situation that you're like, oh, you need a sequencer here. There's a lot of nuances to it. George, anything to add in terms of how automation has helped, especially reducing like hands-on time and minimizing the human error aspect?
Yeah, the tight turnaround times are crucial. They are getting more and more important because we started out providing the NGS testing, we have that information, and now that the clinicians have so much information, the next step will be, so we need it faster because in many cases, timing is very important. So automation is crucial for us to be able to shorten the turnaround times. And the other thing is to make the testing more consistent between experiments because it's automated, so it's every time the same way. It will improve. I mean, people are very important, but it's what it is. I mean, in the morning, the people are fresher in their minds, and so in the afternoons they can be more tired. So some things might go wrong. That's just normal. It's not strange. And so the robot that allows automation in many processes is very important. The other thing also that Bernard was saying is the flexibility. So if we combine an automation with flexibility, that's the best scenario. And what I mean is that we can edit the protocols according to our own particularities in IPATIMUP or any other laboratory. So that we define the protocol and then we have it automated in the machine. So if we have a machine that makes it automatic and we are also able to improve the protocols and design our own protocols, change it, that's the best scenario. It's I mean, since DNA extraction to library preparation to sequencing, I mean, the automation in these processes, it's fundamental.
Yeah, especially of that standardization process, like you said,
That's it.
and having it be equal throughout the day. And humans, like we're great and we're needed, but sometimes it's good to have other standards, and that's why we have protocols and different aspects there. And Bernard, earlier you mentioned that, you know, a lot of your clients at labs are, you know, maybe they start out smaller and then they're growing. Like that is the goal for many labs. From working with so many, what advice would you give for those that do want to scale, but they're worried about maintaining that quality and consistency that George has been highlighting?
Yeah, that's a very good question, Kira. Thank you. We rely a lot on our technology, the MBSeq, and having that technology across all our MGI platform gives us a real peace of mind that we are sure that the quality is consistent across the small throughput to the highest throughput. Okay, so this is the first thing that we always say to the customers. Okay, if you want to scale up, moving from middle throughput to high throughput, the same technology, the quality is going to be the same or even better. And what we actually do as a support team, We try to be with the customer, and part of this is done by validation. We have customer experience centers in Europe and in many other countries where customers can actually bring their samples and do the library prep, run on the sequencer, and see the data by themselves. So I think this is the best approach to work with the customer. They do it with their own hands, they see the data, and they see the quality. And this is basically what we actually do. In case of scaling up, like application, moving from a mid panel to a bigger panel, or even moving from a mid to small throughput to the highest throughput, okay? It doesn't change. We follow up, we stay with the customer throughout this transition.
Yeah. And Nancy, I'd love to get your thoughts more on this different throughputs and experiences that customers can have and how this can be covered with the options that Gene Plus has and can help implement.
Yeah, our solutions, are able to provide the flexible sample throughput based on the customer needs. Yeah, and also based on for different panels. Yeah, for example, we could test one sample, two sample till 48 samples in one run, which I think is very good. Yeah, they can even test the two samples without wasting any reagents. I think this is important for the laboratory. Then they don't need to wait a long time, to combine the samples. So in the future, we are also going to expand the throughput and make it more flexible for the customers. Actually, Gene Plus and MGI work very closely. Yeah, so we'll work together and analyze the needs from the customers and figure out the best automation equipment solutions for the customers. Yeah, so I think from low to medium and to the high throughput. Yes, well, we are very, very flexible for that. Yeah.
Yeah, it really sounds like collaboration, flexibility are the big keys to success in all of this, because those are some of the words, other than all the scientific terms, those are the words that I'm hearing the most from the three of you. But thank you so much for coming on, George, Bernard, Nancy. I mean, it's just been so interesting to hear the process and how all three of you work together so well to really achieve this accurate, fast testing so that at the end of the day, patients can get those results faster, earlier, and, can impact their clinical care. So thank you all so much for weighing in.
Thank you very much. It's been a pleasure.
Yeah, great pleasure. Thank you very much.
Yeah, thank you very much.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.